Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome, everybody, again to another episode of our show, bringing you another really fascinating guest today involved in creating a better tomorrow through their research. Today we have the honor of being joined by Dr. Brandon Berry, who is a postdoc researcher in Matt Kaeberlein's laboratory at University of Washington, and there his research is focusing on the really interesting intersection of aging and metabolism, ultimately how they're linked. And he has, a lot of interest in studying mitochondria, the so-called powerhouses of our cells, and ultimately how they contribute to modulate functional decline that occurs during aging, and ultimately the development of interesting tools that we'll hear about, like optogenetics, to really precisely control these mitochondria and metabolism, and ultimately through these experiments get a better understanding of how mitochondrial dysfunction is a cause or a consequence of various areas of aging, of diseases, and ultimately reveal new ways to understand and impact our health at a higher level. Dr. Berry has an undergraduate degree in biochemistry from SUNY Geneseo. He did his master's and his PhD in physiology, University of Rochester. A lot of really interesting themes to get into today. Really excited about this episode. Dr. Brandon Berry, thank you so much for taking the time to come on the show and talk to us for a little while.
Yeah, thank you. I'm really happy to be here and talk about some cool techniques to think about mitochondria and aging.
Awesome. Yeah, I mean, before we get into any of that, Brandon, I really wanted to hear a little bit more about your background story, about how you got interested in biochemistry, a little bit into the segue into physiology. And, I enjoyed it as I was sort of making my way through your dissertation. I like the little introduction part where you were talking about your time during your master's when you weren't working with mitochondria. You were playing around with zebrafish and eye development. You got pretty good at plumbing fish tanks and all that stuff. Take us back to those days, if you would, a little bit, and how this whole journey got started.
Yeah. So in high school and before, I knew that I was interested in biology. I had some good science teachers. Didn't know if I wanted to go medical or just pure science route. When I was a kid, I was always watching Bill Nye the Science Guy, so I kind of had that predisposition. And yeah, so at the end of high school, didn't really know exactly what I wanted to study, knew I was going to college, and kind of on the common application in New York for state schools, you kind of there's a drop down list. And I saw biochemistry. I thought, I like biology, I'm interested in chemistry. Let's select biochemistry. I didn't know that was going to be a really, really hard major to get through, but I made it through. So that was very fun, learning how to learn differently in college, and kind of cemented my love for basic biology and chemistry. And near the end of my time in college, there was a new faculty, Dr. Travis Bailey, who is still at SUNY Geneseo, and he studied eye development and zebrafish. And I really wanted some research experience because I was starting to think, maybe graduate school, doing a PhD is in my future. So I want to get a little taste of research. So he was new person on campus looking for students, and I was eager to get started. So I joined the Bailey lab, and really because he was just starting and I was kind of just exiting my college career, all of that experience was building a massive tank system. So I got very good with plumber's tape and PVC piping and measuring the salinity and things of the water, and just keeping track of hundreds of zebrafish. So I never got any data from zebrafish, but I really learned how to set up a lab, and specifically how to set up lots and lots of fish tanks. So that was very fun and interesting. And then the other interesting thing about during college was I was in high school, and before I was always playing music. So I was in punk bands with my friends in high school, and during college we had a snow day, and I needed to do something for extra credit in my biochemistry course. And I thought, there were a lot of like ways to memorize biological pathways like the TCA cycle and glycolysis. And I was like, I'm going to write a song to memorize these things. I'm going to show it to the teacher and maybe get some extra credit from it. So I did that, and I actually forced my band into recording the song. So somewhere out there is a college song version of the TCA cycle. And I always put that into my my graduate school application letters, and it's something that I think stood out a little bit that helped me get through the hardcore science of it all.
Excellent. And as I mentioned, Brandon, in the bio, you have been, per your master's work into your PhD, heavily focused on mitochondria and mitochondrial metabolism, and obviously mitochondria is something that we've mentioned a bit on the show in the past. I think most viewers and listeners will know sort of the two themes that, yes, we have these mitochondria in our cells. They are our powerhouses. They provide us with energy, and they make this ATP stuff, the energy, the currency of life. We're not going to go into that. I think people have heard that story a lot, and most of our listeners have been through sort of high school biology. Where I do want to go is to a lot of the really unique themes that you touch on, actually before and into your PhD, and now some of the stuff you're working on. And I really would like to spend some time in the sort of the 2020 timeframe. You do your PhD. It's entitled Optogenetic Control of Mitochondria, Alter Cellular Energy Sensing, and Hypoxia Resistance. And I encourage everybody listening and watching to go out, take a look at it. It's really a very impressive piece of work. Three really important themes that I'd like to go into that you highlight in your dissertation. One, that beyond that organelle, mitochondria being there to provide us with that energy, it is a really complex organelle that has really unique dynamics flowing through its membrane, something known as the proton motive force that I'll like you to take us into, that provides ATP, that provides sort of this energy sensing capability. You highlight the really interesting concept as well that we don't normally think of, the spatio-temporal dynamics of mitochondria. They're floating around in the cell and they're doing their thing, but really they have some unique positional timing component as well as just pumping out the ATP. And then obviously the third theme, the optogenetics, the ability to manipulate this unique organelle with light. Let's go to the first one first. Take us into the unique membrane dynamics of the mitochondria, what the proton motive force is all about, themes that we don't, I think, normally think about as much when we hear about the mitochondria, but extremely important in your work and where your work is going.
Yeah, definitely. So yeah, this proton motive force is an electrochemical proton gradient. So it's an electrical component and a chemical component. So there are two potential energies. So lots of people probably know mitochondria have two membranes. We're talking about a gradient across the inside membrane. So the inside membrane is pretty impermeable. Nothing can get across unless something in that membrane lets it across. So how this electrochemical gradient gets established is very much like combustion. So this is why the whole powerhouse of the cell analogy came about, is because it's very similar to a coal burning power plant. It's a carbon source that gets combusted, and that energy is stored and transformed somehow. So the coal is food and the metabolites from food. So we digest the food. Combustion happens in the inner membrane of mitochondria with the enzymes of the respiratory chain. So oxygen gets reduced, CO2 is produced, just like it is combustion. And how the energy gets captured is protons, positively charged hydrogen ions, are pumped from inside the mitochondria to the outside. And they establish this, what I like to call the cellular battery charge, because it's like a battery, positive outside, negative inside. So positively charged things in the cell will feel an attraction to inside the mitochondria because they got a negative charge inside. So those protons are subject to that gradient. And the enzyme that ends up making the ATP lets the protons go down their gradient, and it releases that energy, and ATP gets made from ADP. So that's, core powerhouse of the cell analogy. And kind of like you said, that's where the high school education of it all ends, and that's why everyone knows mitochondria are powerhouse of the cell. But what everyone doesn't really know is that this proton gradient does a lot more than just help the mitochondria make ATP. It fluctuates. So if you watch it for a long time, it will go up and down in kind of stochastic ways, as far as we can tell. Kind of unclear why or how that's happening. And this kind of gets into the spatial temporal stuff, which we can get into a little more when that comes up. But it does a lot more, like energy sensing, which we'll get into as well. And it can look different in different tissue types. So the mitochondria in skin cells, for example, we have mitochondria in our skin. Energetically, it's kind of unclear, like what's using energy in our skin cells right now compared to something like the heart or the brain. Right. It's a little more obvious that we're using lots of energy in our heart and brain. So I was kind of fascinated by metabolism and mitochondria. The whole powerhouse analogy got me interested in mitochondria. And actually in college, we isolated mitochondria from cauliflower. And it was something that was a real turning point for me. We kind of shaved the tips of cauliflower off, and it's white. And as we go through the steps of isolating the mitochondria, eventually the sample started turning darker and darker, and it was like a brown red. And it's like, that's mitochondria. There's light absorbing things in there. So to see the white tissue from cauliflower turn into this mitochondrial color was kind of amazing. And then putting it into the instrument that measures oxygen, seeing them consume oxygen, it was a moment of like, wow, these things are real inside of our cells, and they do what we were taught they do. And that's kind of amazing. And then going into graduate school and learning about the real nuances of like, they are powerhouses, but they are a lot more than powerhouses. There's an idea out there now that people want to start saying that they are the processors of the cell, sort of information processors. So they know how and when to provide energy, and they're not just spitting out ATP willy-nilly. Yeah.
And then, as you were just mentioning, this is a nice little place to add in the spatiotemporal component, and you were just mentioning about, a cardiomyocyte versus a nerve versus whatever. The mitochondrial, what we'll say, dynamics that go on and we have obviously 200 plus different cell lineages. One that's always been kind of interesting to me is just the oocyte and embryogenesis, about how mitochondria sort of is I don't know if the term is swims around or is thrown around as the whole process of life begins. Talk a little bit about some of what, along the way you've learned as you've looked at these different cell types, and we'll go into some of your later papers as we get into the themes of different diseases. But what have you learned in terms of the spatiotemporal dynamics of how mitochondria is used, it fluctuates, moves around these cells as it does its daily activity?
Yeah. I've got a few really fun examples about that. So going back to skin cells, it turns out that mitochondria in skin cells, or fibroblasts, look very much like a plate of spaghetti. They're all super long and intertangled and kind of disorganized looking. And when you look at mitochondria in a muscle cell, it's like a grid. It's very grid-like, very regular, and they have very regular protrusions. And compared to something like a liver cell or a kidney cell, where mitochondria maybe look more like that textbook image of the bean-shaped, discrete organelles. So just right off the bat, in different places in the body, mitochondria look very different. And how they look is very tightly correlated with what they're doing for that cell. So for example, in something like a neuron, we've got small mitochondria that can travel down the axon of the neuron or travel back to the cell body. to go where the energy is needed to let neurotransmitters be released. And the mitochondria in the cell body of the neuron that are kind of helping with gene expression are around the nucleus and look different than the mobile mitochondria. So these are organelles that are really responding rapidly to the cellular environment, and they are causing the cellular environment to change rapidly. So this is the cool thing about mitochondria and also the difficult thing to study, because once you start trying to manipulate something about the cell or something about mitochondria with traditional methods like drugs or genetics, it becomes pretty hard to tell, is what we did causing mitochondria to change, or is there a mitochondrial change that's causing something to change in the cell? So it's a double arrow, and sometimes it is a double arrow. There's feedback in both directions. So when you want to do careful causal science, it can be hard to interpret what's going on, especially when you're looking in different cell types.
Right.
So to kind of take a really 1,000-foot view, I'm always thinking about, can we achieve mitochondrial medicine? So like you said, people know what mitochondria do at this high level. They're the powerhouse of the cell. High schoolers learn this. Why is it then that we can't go to the doctor and get something to treat our mitochondrial problems? And the biggest thing is we just don't know enough about how diverse mitochondria are, and we don't know enough about what mitochondria are actually doing other than just providing that energy. So you can imagine a situation where you study skin cells, let's say, and we have a mitochondrial intervention, cells in a dish. That's great. We have amazing effects. You give it to a person and it kills the heart cell mitochondria. That's something that's just not a non-starter. That's not how we should be approaching mitochondrial medicine. So I think what I'm really passionate about is taking what we know about the basic biology of mitochondria in things like cells and worms and flies and these like very sciencey things, and asking, what can we learn about tissue in a human being from those things? What are the fundamentals that will end up influencing the human biology so we know what to go after specifically? And I've kind of made my scientific career about focusing on this one aspect, proton motive force, PMF, cellular battery charge, because it's such an integrator of all of the signals that mitochondria receive and put out. Everything kind of feeds into this central charge that can talk to a lot of different things. So I think, if not intervening on the charge, kind of monitoring the charge in humans would be something really amazing. And I don't think we're there yet, but that's kind of my approach to the diversity of mitochondria. Is they've all got this gradient, and what can it tell us?
And the tool, as you know, mentioning, there's been cruder, older methods, as you were saying, whether it's drugs or what have you, to try to manipulate mitochondria in the past. You've chosen sort of one of the more cutting edge technologies that's out there today, namely optogenetics, the use of light to ultimately impact these. Whether I think you specifically are focusing on the light activated proton pumps that are involved in the PMF, and optogenetics is obviously being explored to as a really cutting edge way, let's say, to hit these ion channels, the pumps, the enzymes, whatever that are associated at that level. Say a few words about this, if you would, because optogenetics we don't talk as much about, but it really is, I'll say, a bleeding edge tool that we're seeing more and more of in lieu of sort of older, cruder approaches.
Yeah. So optogenetics, just broadly, is the use of some light-sensitive protein expressed in a new system that usually doesn't have light-sensitive proteins. So then you can use light to control some aspect of physiology. And like you said, we knew we were dealing with a proton gradient in the inner membrane of mitochondria, and we knew that there were light-sensitive proton pumps, so these enzymes that can move protons across a membrane. So for my advisor during graduate school, Dr. Andrew Matovich, to him it was kind of clear, like, we have an opportunity here to take one of these light-sensitive proteins and stick it into mitochondria, and then we can control this gradient that is so integrative of all of these signals. Like, what if we could just, maybe stop that integration by just controlling it as an independent variable? something that was really hard to do previously. Really, the only way you can control that gradient is using drugs that totally collapse the proton motive force. So it's got a big off switch or nothing. So we wanted to have a little bit more nuance and maybe give it an on switch. And that's why we kind of called this tool mitochondria on, or mito on is how I'll refer to it. So yeah, we took this light activated proton pump from a fungal organism that normally would use this proton pump to sense light and to move toward the light to do photosynthesis. So it kind of has a very predictable role in this organism. So we thought, let's clone that gene, stick it into mitochondria using some molecular tricks, and then we can control mitochondria by turning on and off a light. And the other good thing about optogenetics is that this light sensitivity can be very precise and reversible. So you can shine a laser in a specific spot that you want it and no light elsewhere, or you can turn the light on and then off at different times to kind of start dissecting the temporal aspect of, turn it up now, then let it be. So that was a long road at the beginning of graduate school, getting the right proton pump. We had a few options, and we had lots of options of ways to get this into mitochondria. And we didn't really know which was going to work because it turns out if you stick some foreign thing into mitochondria, they're not very happy about that. They can try to get rid of it, or they're very sick. So we had to do some work to kind of find a Goldilocks amount, and using different molecular tricks to make animals. So we did this in a worm model, and it took a little bit of trying to actually get normal, healthy worms that had this and that was functional. So yeah, optogenetics has kind of revolutionized neuroscience a little bit with causing neurons to fire. So neurons use electrochemical gradients across their cell membranes. So it was kind of a very obvious approach to shine light on brains. You can see there are mice that have fiber optics going into their brains. You can turn on the light and they'll get very aggressive, and it's all very cool. And we kind of thought, let's do that, but with metabolism. And yeah, it was frustrating at first, and then it got very fun once it started to work.
Yeah, that's it's an amazing, set of, well, it's an amazing piece of work. And I wanted sorry if I took too much time focusing on it, but I really wanted to, at least for the audience, to outline the way you think about, not just sort of this mitochondria 2.0, but obviously bringing in these other tools, and again, thinking of the physics as well, when we as again bring in the spatial temporal dynamics to all this. So really a great piece of work. From that, laying that groundwork, let's go into some of the areas that you're researching now in terms of human health and some of the applications of these tools. One that actually you started thinking about it actually as you were doing your PhD, and you've published since then. So you published in 2018, Optogenetic Regulation of T-cell Metabolism in the Tumor Microenvironment. More recently, Optical Control of CD8 T-cell Metabolism and Effector Functions. And you highlight here, this really interesting interaction. Here we are in the age of immuno-oncology. We normally have sort of a microenvironment where, our immune system is fighting cancer, and there's sort of this back and forth. Now we bring in this third component, sort of the immuno-oncology world of adding T-cell therapeutics. And you talk about in the recent paper sort of this fierce, what you say, metabolic competition that's going on between all these cells, and ultimately, talking about how, per some of the tools you were just discussing and concepts, you can ultimately help boost or enhance a metabolic advantage in sort of the T cells that we want to help kill these cancer cells off with, and ultimately give this advantage when we want to destroy malignant cells in various different types of solid tumors. Take us into this, because I think this is a really elegant way to think about not just obviously the mitochondrial manipulation, but ultimately how that translates up to cells in the broader cellular microenvironment.
Yeah, this is a really fun and interesting story of how we got involved in this T cell world. So the lab that I developed these tools in, kind of like I said, was a C. elegans lab. C. elegans are tiny worms that live in the soil. They're transparent. Lots of basic biology is known about these worms. It was kind of a good system to characterize it. At the same time, there was another optogenetics lab at the University of Rochester, a lab run by Dr. Min-Soo Kim, and his graduate student at the time, Andrea Amatrano. I think her name is Andrea Manel now. She kind of led this paper, and their lab did optogenetic approaches in T cells, immunology, and cancer immunology. So they had some other optogenetic approaches to kind of control T cell metabolism. Now, my PhD work was on hypoxia. So we were exposing worms to hypoxia as a heart attack stroke model. We were thinking about low oxygen, mitochondria use oxygen. Can we use light to bypass the need for oxygen? So we met with this lab who was doing kind of a very similar thing. One of the metabolic challenges in the tumor microenvironment is that it becomes very hypoxic. Tumor, especially solid tumors, get very hypoxic. They use up oxygen. Oxygen can't penetrate the tumor. So they were kind of thinking similarly that part of the metabolic challenge is low oxygen. Can we use this mitochondrial tool to kind of say, you don't need oxygen anymore. We're going to charge up the battery with light. So we, again, spent some time adapting the worm molecular biology stuff to get Mito-on, this light-activated proton pump, into mouse T cells, into human cells in culture. And it, again, after some trying, we got that to work. And Andrea was able to kind of beautifully show that in a simulated tumor microenvironment, these cells were able to move for longer. They moved differently. It seemed like they were moving in a more healthy way, be able to infiltrate cancer cells better. And they were producing more cytokines, so more signals to kill the cancer cells. And it seemed like the cancer cells in culture, so in vitro, were actually able to be killed more easily. So that was extremely exciting, and they're still working on kind of getting this into a more in vivo model to test this. And I'm really excited by that because it really is I say real life application for this thing. Like science and having a good scientific tool in worms is also real life, but this is a kind of shows me a proof of concept that we can actually affect human biology and something that is like actually happening, such as T cell therapy. So yeah, that was a really fun collaboration, and I'm excited to see what they're going to come out with next.
Yeah, no, I was equally excited about it. Again, we hear obviously we're in the early days of immuno-oncology and CAR T and so forth, and we hear about we have these successes, but we need other other strategies to go beyond, to go into solid tumors. And the fact that this was not just another strategy, but one that again uses all these really cutting edge tools, I thought was really exciting. So I appreciate you walking through that. Another piece of this, and I guess this also brings in an interesting microenvironment and genetic, both topics. But you recently, just a couple of months ago, wrote in Nature Reviews on Endocrinology, a mitochondrial origin for inherited diabetes mellitus. And here again, you're talking a little bit about the models, but at the same time, as you were just saying, how we go from understanding what's happening in vitro to the uniqueness of the in vivo world. And here again, we have a disease that has a genetic underpinning, but then, spreads out from there and has its own unique microenvironmental metabolic challenges. Talk a little bit about what you are interested in and you foresee specifically as it pertains to endocrinology and specifically in this case, T1D.
Yeah. So I think about this kind of metabolic diseases all the time and the sort of translational aspect of what I'm doing. So I'm mostly use worms, a little bit of mice, but I think if it doesn't end up translating to a human situation, then it's kind of it's got to be very clear why we're doing what we're doing. So I always try to make sure that what we're studying is something that has a good promise to do that translation. And I think mitochondria are a great organelle to study because it's just clearer and clearer the more you read the literature through a mitochondrial lens that mitochondria are involved in every pathology and disease in some way. And it kind of makes sense if you think about every action that a cell takes. Anything that happens in a cell is going to be using energy. Anything that is moving in a direction, any choice that a cell is able to make uses ATP. And most of the time that ATP comes from mitochondria. So if you have mitochondria that are dysfunctional in some way, of course, there's going to be some cellular effect. So in all these diseases, of course, at some level, mitochondria may not be the cause of the disease, but there's going to be an energetic defect at some level in any pathology. And the most obvious pathologies are in endocrinology and metabolism. Mitochondria are hubs of metabolism.
Yep.
So type 2 diabetes and type 1 diabetes as well, this very interesting case from the 90s that I wrote about in Nature Reviews Endocrinology was just a striking example of a specific mutation or deletion in the mitochondrial genome. So mitochondria have their own DNA. They came from an ancient bacteria-like organism that got engulfed into eukaryotic cells. So we're kind of carrying around these little visitors in our cell that have become part of us. You change one aspect of their DNA and you have an inherited form of diabetes and deafness. And that's not obvious from the symptoms. So you're unable to hear, you are unable to handle glucose. Maybe that's not obvious that it came from a single event in mitochondria. So one thing that happens in these tiny bacteria in our cells results in a very complex physiology. So I wanted to cover that for their journal club series to kind of show that a very complicated process can be boiled down to a single problem in mitochondria. Now, it's unclear. We can't just go in and fix that single problem and fix the individual. But why is that? I think that's a good pathway to be on. And from that, so starting from a human disorder, a human problem with mitochondria, then you can take it back to animals and say, what can we learn using these tools and interventions that we can do with animals, and then kind of bounce it back to the human condition. So I think, keeping that the translation lines open and always thinking about, when you're working with the animals, you're thinking about the humans. When you're working with the humans, you're thinking about how you can figure more out in the animals. So I thought that was a great way to start that line of thinking for some people. And there have been papers like that in the past that kind of were aha moments for me, like, oh yeah, like clearly this is a basic biology problem that is manifesting in humans, and we can, start to think about how to do something about it.
And then, Brendan, moving from, we talked cancer, we've talked diabetes. You and Matt and some other folks as well have been publishing recently, last couple of years, on sort of what you define as sort of an energetics perspective on geroscience, thinking about aging broadly. And again, coming back to sort of the older tools or what's been proposed, things like shuffling around mitochondrial genes or mitochondrial transplants, things like that. I have my own perspective on where all that might be going, but ultimately, again, thinking of the tools that you're working with, thinking of some of what you and Matt have been focusing on in terms of aging broadly and PMF and the mitochondria. What are some of your strategies in moving forward with let's call energetics gerosciences? Where do you want to take this? Here we are, 2023. You've done some work on I believe in C. I think it was in C. elegans, but take us a little through how you think broadly about aging per the story of energetics and optogenetics.
Yeah. So I'm going to start at the end of grad school, where I was doing this hypoxia stuff.
Yep.
And I kind of learned that a lot of the interventions that we do in especially the worm models of hypoxia and a lot of the mouse models of heart attack and stroke, there are some powerful interventions that can be very protective. And a lot of those interventions stop working the older the animal gets.
Okay.
So my thought process was, why are we studying things that don't work in the population that is going to need this intervention. Old people have heart attacks, old people have strokes, and we're studying interventions that don't work in older animals.
Yeah.
And so that's what got me into aging. I was thinking, aging biology must be pretty important if these very powerful effects go away in an aged biology system.
Mm-hmm.
So I reached out to Matt Kaeberlein. I really liked Matt's lab because of his breadth of models. They used yeast, worms, mice, dogs, and he had some mitochondria expertise as well. So I thought, who better to learn aging from and be able to do mitochondria science with. So very early on, we wrote that review article, an energetics perspective on geroscience. So geroscience is this idea that if you target the basic biology of aging, is what happens to an older cell, it will simultaneously target lots of dysfunctions that are associated with aging and lots of age-related disease. And kind of like I alluded to, the more I read about aging and the so-called hallmarks of aging, these are 9 hallmarks that are very well investigated. I kind of started to see, like, from my mitochondrial lens or from my maybe mitochondrial bias, that a lot of what's going on in the hallmarks of aging is an energetic problem. And maybe if you target mitochondria, all of the other hallmarks will get targeted as well. And that, again, kind of makes sense because all of the other hallmarks are using ATP in some capacity. So, mitochondria are connected to all of the other hallmarks. Is mitochondrial dysfunction the, core of the hallmarks of aging? I think it might be. That's not to say that you can't target the other hallmarks as well. But I think targeting mitochondria might be a way to, help with everything else. So that's kind of what I was operating under when I joined Matt's lab. We wrote that paper together. And then I started to notice that this very specific thing that I study, the proton motive force, everywhere that I found in an aging biology paper that actually measured that variable well, it seemed to be declining with age. And this is how I started to develop this battery charge analogy. I was like, okay, this is like a laptop battery where you have charge and drain cycles, and eventually that battery is not going to hold a charge as much or for as long. It's not going to recharge as much. And I thought, I really like this analogy. It's not a perfect analogy. Obviously, mitochondria are a lot more complicated than a laptop battery. But it seemed to hold true that anywhere you look in aged tissue, mice, worms, yeast, very highly evolutionarily conserved, aging seems to correlate with this decline in the battery charge. So I thought, is something about aging telling mitochondria to not be as charged, or are mitochondria just losing their charge and that is what's causing aging? That's kind of a simplistic way to think about it, but our models are simple, and Matt will always say that your model is wrong. So we need to find out how the model is wrong. So I had this optogenetic tool that directly could charge up this battery charge. So I thought, great, let's charge the battery and see what happens in the worms. Worms are very easy to measure their lifespan. We had these worms ready to go. So what I did was waited for the worms to start to lose that battery charge. And they start losing the charge very early in life. So we can start an adulthood intervention, but they're still pretty healthy. So we illuminate these worms, charging their mitochondria for their whole life. And kind of amazingly, they lived about 30% longer than population without this intervention. Didn't believe it on the first go. I had never really done worm lifespans before. And I was like, okay, I don't know about this. So we took it to Matt's lab, learned how to do good worm lifespans. We included so many controls. And after the paper was under peer review, we included even more controls to really convince ourselves charging up this mitochondrial battery is extending lifespan in these worms. And that's what we found, and we published that, and we're pretty happy with it. But it still begs the question: why does it extend lifespan? Why does just rescuing this mitochondrial decline extend the lifespan of the worm? So it suggests that mitochondrial charge specifically is a pretty powerful regulator of aging, at least in these worms. And so part of what we want to do is test it elsewhere, make sure that this is not a specific worm mitochondria thing. So this is underway in some other organisms now. And then downstream from that, we want to know what are the cellular components that are reacting to this maintained charge with age. I kind of know that it's not going to be one thing that is responding, and sometimes that's very hard to get funding to study and to publish, because a lot of times you want, this is the pathway that regulates X. And when you mess with something as fundamental as the cellular battery charge, there is no the pathway. There's probably lots of pathways. So we are kind of going in our small group, one by one down the pathways that seem likely to be responsive to this mitochondrial battery charge. But there are lots and lots of things to test. And I'm really hopeful, because every time I talk about this at conferences or whatever, everyone has a different idea how to use Mito-on to study what they think is the important thing to study. And I'm always just like, yeah, we haven't thought about that. We haven't done that. You should do it. Let me send you the plasmid. So I'm really excited that this is getting out there. More people are using it. And that is kind of how I think about just the basic biology aging part of it. That's how I came to it. That's how I think about it, with this very mitocentric view of it all. That being said, like I said, I'm always kind of thinking translationally as well. And I think a lot, and Matt has asked me a lot as well, like, how do we measure this in a human being? And I don't know, there's not a good answer to that, because to get at mitochondria in a person, the best way right now that we have to do this very translationally is to take a punch of your leg muscle out and prepare it and study it in isolation. And, mitochondria don't look like they do in your body after you punch it out of your leg and put it in, laboratory conditions. So it's like, we can get a sense for, okay, you have this piece of muscle. You can ask, how charged can these mitochondria get? How decharged can they get? How much oxygen can they consume? But it doesn't answer the question, what was the charge when it was in your leg? And that's just your leg muscle. What is the charge in your kidney mitochondria right now? What is the charge in your skin mitochondria right now? There's not a really, robust quantitative way to get that answer. There have been encouraging papers recently. I have a name written down here, I think. I can send a link to this paper where they actually measured this in vivo in humans using a relatively new method, using PET scanning and a tracer molecule. And they did this in a lung cancer model. And they were able to show that within specific tumors, there are mitochondria that have a higher battery charge, and there are mitochondria that have a lower battery charge. And they kind of characterize why that might be. But it was in vivo, in a person, in a mouse. They can do this. I think they've also done it in some pigs. So I'm pretty excited by that technique of actually looking in a living animal. Now, there have been some responses to these papers saying, like, well, this isn't very quantitative. To really know, you need to do it better. And it's like, yeah, you do need to do it better, but also to do it better, you need to, take the tissue out. And then at that point, it's hard. So I think, thinking more translationally, that there are only going to be more methods to look at this in vivo and more scenarios where it's going to get easier to look at this in a human. So I am hopeful for that. And, that makes me hopeful that there will be, true mitochondrial medicine that will be able to get a mitochondrial diagnosis and do something about it that's outside of just pure mitochondrial disease. So mitochondrial disease, like genetic diseases, we know some things about that, but, getting your mitochondria treated if you have diabetes, we don't know how to do that.
Got it. Yeah, I mean, look, you're really on the cutting edge of all this stuff, and you got a lot to explore. So, anyone that ever says, whatever, it's take it with a grain of salt and keep going forward on this one, because I mean, you're really you have so much to learn and so many possibilities as it pertains to bioenergetics and health. It's just it's going to be an exciting coming decade for you. The one thing you made me think about, because when I was talking to Matt, obviously, as you were just mentioning, a lot of different model organisms there, and thinking back to sort of what we started out with here, and you're plumbing the zebrafish tanks. I've noticed a lot of work, obviously, merging optogenetics and zebrafish in the last couple of years. Any sort of connection there back to, I know the zebrafish fits in somewhere there between C. elegans and the mice and whatever, but obviously a really cool model, a lot of interesting things on the regenerative medicine front there. You doing any zebrafish things with the
Yeah, I'm not.
optogenetics?
Yeah,
If you
so I'm
can't
not
talk.
personally doing anything with fish, but there are people interested. Not specifically in zebrafish yet with Mito-on, maybe for things like regeneration, but for aging, there are some people interested in some shorter lived fish. It turns out that zebrafish
uh,
live quite a long time,
yeah, yeah,
but there are fish that live shorter. So on the aging side, there are some people interested in that. So I'm hoping to see it in fish very soon. Um, and again, the great thing about fish is that they're relatively transparent, so you can
yeah,
actually get the light in them. Especially like zebrafish are definitely transparent. Again, harder to do lifespan there, but yeah, and like some of the optogenetics that's already happening in zebrafish was kind of inspirational for that approach as well. So
yeah.
um, yeah, I'm
You know,
hopeful for
I
that
was just
stuff with
thinking
this.
about, the, as you were saying, the regenerative component about, I love the sort of the regenerative side of aging. And, they always talk about, when you talk about iPS cells and sort of nowadays, you always talk about, oh, well, mitochondria gets remodeled along the way. And it'd be interesting to see, how mitochondria is involved in some of that sort of regenerative dynamic, how, spatial temporal dynamics of mitochondria affect regeneration. But anyway, that's for a whole other set of projects. But it's
Yeah, yeah.
really, really great stuff, Brandon. I mean, I'm just so impressed hearing everything you've done and where you're going with it. Anything else hot for 2023 that we should know about in terms of maybe conferences you're going to be attending or presenting at, places that we can run into you, meet you, talk to you? Anything else hot on the calendar or the lab calendar that we should
Yeah,
know
yeah.
about? Please.
Yeah, so I'm going to be at the Fusion Mitochondria Conference in Portugal in July. Just got that registered. So I'll be there talking about this stuff and some other stuff. I'm on Twitter at MitoBrandon. So I'm always, tweeting about all things mitochondria. I'm working on getting a little bit of a newsletter blog thing going and maybe some audio content in the future talking about weird and wild mitochondria. The animal kingdom has some extreme examples of very cool mitochondria, and I think talking about that would be very fun. Um, so those are the more public things. And then I'll also say that I'm really excited for things like Matt's Dog Aging Project and more big projects that are going to start taking the environment more into account.
Okay.
So lots of science is done in a very controlled, germ-free environment where we control all the variables. And I really think for human mitochondrial medicine, mitochondria, one of their roles is to help deal with toxins. So I think there's a lot that we still don't know about how mitochondria respond to different environments. So you
Mm-hmm.
have someone living in a very polluted city, and you have someone genetically identical, living in a, paradise nature place. Their mitochondria are almost certainly going to be very different. And I think starting to capture that diversity is going to become a bigger thing, especially in aging research.
Right.
And, it's kind of, as a scientist, maybe can make you uncomfortable of like, oh, we need to control all those variables to really learn about the biology. But at the end of the day, you have to think, are we interested in mouse biology or are we interested in health?
Yeah.
And part of health is having very diverse environments. So I'm excited to start seeing more people consider, hardcore mitochondrial biology and the interaction with diverse environments and toxic environments. Because there are a lot of toxic environments out there that
That's true.
are certainly going to interact with metabolism and mitochondria. So I don't think mitochondrial medicine will really be achieved until that is also being accounted for.
Got it.
Um,
Got it. That's a whole
and
area.
yeah, I'd also like to plug the United Mitochondrial Disease Foundation is a great foundation that's doing basic science mitochondrial work focused on mitochondrial diseases. And the Michael J. Fox Foundation for Parkinson's Disease also funds lots of mitochondrial research. So those are places that, if people want to have an impact on mitochondrial science that is sure to impact humans, those are places to go, along with places like the American Heart Association, are places that I'm always looking to as well for my mitochondria related stuff.
Outstanding.
Yeah.
Really outstanding. Great work, Brandon. I mean, as I said,
Thank you.
Wishing you the best with all this. I'm excited for you, especially
Thank you.
when I see combining the cutting edge technologies with really sort of more of an I don't call it unexplored, but a domain that is less traveled, and wishing you the best with all of it. For everybody that's going to be listening to this particular episode of our show across the podcast networks or watching on the YouTube channel, again, you've been spending time with Dr. Brandon Berry, postdoc researcher at the Kaeberlein Laboratory, University of Washington, doing really amazing things at the intersection of metabolism, energetics, aging, optogenetics. Brandon, I want to thank you again for taking the time out of your schedule to come talk to us for a little while and educate us on these really interesting topics. Obviously, thank you for what you do. And as I like to say here on our show, thanks for helping to create a better tomorrow for a lot of people via what you do. Really great story.
Thank you so much. Thank you for having me. I'm happy to be a part of such a great show.
Great having you.